Okay, so hopefully that's good to go. All right, so welcome to the Lunch and Learn for December. We are today we're going to talk about. Let me just make sure I've got. There we go. Okay. We're going to talk about spoilage yeast, and I try and schedule these lunch and learns to an appropriate topic, to something that we might be thinking about at least in our wineries at this time. And so I thought spoilage yeast is something that we are trying to prevent, and maybe a timely topic as we're probably done our fermentations. We are maybe starting to work on fining and filtration and whatnot and thinking about packaging. And so this is what we're really trying to avoid and prevent these guys from growing and creating any issues in your wine quality. So let's jump in here. So what are yeast? And I'm going to focus mainly on yeast today. There's lots of bacteria as well that are spoilage bacteria, but I like to kind of condense it down into a smaller topic. So I am going to focus mainly on yeast for this presentation. So they are part of the fungi kingdom, and this includes both yeast and mold. So there's a couple molds I will mention as we go through, but they're all lumped together. They are eukaryotic cells, okay? So they're actually much more closely related to us and our cells than a bacterial cell. So just keep that in mind. They have a lot of the same cellular processes that that our own bodies perform, like um, uh, glycolysis, like oxidative phosphorylation, and all of those metabolism type of functions, as well as a lot of the organelles that they have, the nucleus, the if you remember your high school biology, the Golgi bodies, the endoplasmic reticulum, they've got all of those things, whereas bacteria have some of them for sure, but they are very different, let's say. So we can't talk about yeast without at least mentioning Saccharomyces cerevisiae, because for the most part, Saccharomyces cerevisiae is our friend. It's what we typically use for our alcohol-producing yeast. A lot of strains, and we do use specific strains, right, when we're winemaking, but a lot of the strains have favorable aromas and flavors, and they've been selected for those favorable aromas and flavors, as well as their high alcohol production. Wine strains have high tolerance to ethanol and a high tolerance to our low pHs, right? Our pH between 3 and 4. Our wine yeast strains are going to survive and ferment. And they're as an aside, they're facultative anaerobes. So what does that mean? They will produce alcohol even in the presence of oxygen. Okay, so they even if oxygen is present, they will prefer to use the mechanisms, and I'm not going to go through the mechanisms, but they will go through that mechanism to produce alcohol rather than oxidative phosphorylation, which is what our bodies do. And the other thing I want to mention too, when we're talking about Saccharomyces cerevisiae, it can be a spoilage yeast. It's yeast spoilage. If you're calling something a spoilage yeast, it's kind of like calling a plant a weed. It's a weed if you don't want it. Okay. Otherwise, it's just a plant. So Saccharomyces cerevisiae, a good example of this would be if you have some sugar left in your wine and you have a Saccharomyces cerevisiae infection, maybe it's in bottle at that point as well, you're going to get a fermentation going on, just like a normal fermentation, but it's not necessarily what you wanted. And then you might have gushing and popping corks or things like that. And so that's spoilage. So we can control that, and I'll talk about it at the end of today. We can control if we don't want that to happen. Okay, so a spoilage yeast is just something we don't want to be there, because there are other yeasts we could use for fermentation. I'm not going to get into those today, but there's lots of interesting research and whatnot going on with alternative yeast strains and even species. So, what is a spoilage yeast? It's an undesirable yeast strain that causes negative effects to a wine. Okay, doesn't have to just be aromas and tastes. It could be, that you got a secondary or a fermentation, a secondary fermentation happening in your bottle, let's say. Often they are non Saccharomyces species. So the ones that I'll show you today are non-Sac species. But doesn't have to be. I'm not going to cover all of the potential spoilage yeast because there's lots and lots out there that could infect your wine, but I will talk about the most prevalent ones. So they can impart flaws or faults that are perceived on the aroma, and that's how a lot of the faults are perceived. A lot of the but you can also have visual faults. Some of them cause hazes, especially if they grow in large numbers. You're definitely going to start to see haze in your bottle. Taste, they can alter taste because a lot of them can metabolize sugars or metabolize acids, and finish as well if there's some unwanted, bitterness or things like that due to compounds that they produce. Infection can be caused in the winery, tank or barrel, or in the bottle. And I should say this as well, and you can have problems on the grapes as well. So right from the vineyard, you can bring those through the winery, and then they can impart negative faults into your finished wine. So you can really, at any point during the growing season or the winemaking, you can get these infections. And I think most people would agree with this, but prevention is really the key here. It's a lot easier to avoid these problems than to deal with them once they're in your wine. Really hard to get rid of aromas and visual faults and things like that. There are a couple things you could do, and I'll mention them, but trust me, you don't want to be wasting your time doing them. So we're going to jump right in. I'm going to show you a handful of the common spoilage yeasts that we see and what they do, how they cause, or what they cause fault-wise in the wine. I don't expect you in your wineries to be identifying exactly what types of infections you have. If you know you have something wrong, you can always send it to us and we can put it under the microscope. But there are a couple of techniques I'll show you at the end of the first step you should take immediately if you think there's an infection. But I do think you should know some of the species that are common and what they can do to wine. And some of them are you'll see why I say, I think by the end, why it's not always necessary to ID exactly what yeast you have. Sometimes it is important, but not always. So Brettanomyces bruxinensis, that's not how you say it. I'm going to leave that alone. Also known as Decara. And so there, a lot of times, the reason I'm putting up the two names, and it has to do with reproductive forms of the yeast. You may come across it in a textbook or an article. You may come across both names. So just Google what the other one is or Google it if you think it sounds weird and you can't place it. So the cell description for Brett, and this yeast causes a Brett infection. Spheroidal to ellipsoidal, you'll see in the picture they're a little bit more ellipsoidal than what Saccharomyces cerevisiae usually is. Saccharomyces is usually just a little bit ellipsoidal to spheroidal. They reproduce by multilateral budding, meaning that they'll bud off of any point. Oh, you can't see there we go. They'll bud off at any point along the cell. So it doesn't happen just at the ends, although it kind of looks like it in this picture, but it can bud off of the sides as well. They form pseudomycelium, so they can form films. And there's a lot of these yeasts that will form films on the top, on the surface of your wines. So wine spoilage, what do they do to your wines? They under aerobic conditions, so they need some oxygen to create acetic acid, so they can metabolize glucose and form acetic acid. And there's also the production of 4-ethylphenol, which is that Band-Aid smell, or 4-ethyl glycol. So clove, which sounds not so bad, but it usually is in high concentration, so it gets into that that smokiness. And the issue with Brett is that it's really resilient and it's ubiquitous. One place that Brett loves to live is in barrels. So if you are reusing barrels over and over, wood is a really bad uh, container for wine, and because of all the nooks and crannies, it's not a very nice smooth surface that we can clean easily. And bacteria and yeast love to live in those nooks and crannies, and it's moist, it's humid in a barrel cellar as well. So yeah, barrels are just not the greatest, but because traditionally they've been used for so long, I don't think they're going anywhere. But if you do have a Brett infection in one of your barrels, find out how to treat it appropriately if you are not going to get rid of it. And that could be steam or other cleaners. I'm not going to go through in detail other cleaners and things like that today, but I just warn you that they can live for a long time and you'll probably have infections down the road. The other thing I would highly recommend, let's say you're taking a sample out of your barrels and you have a wine thief, or you're just using a
One moment, everyone. It seems that we lost Erin. I'm going to have her hop onto my computer here real quick. So just give me one moment here. So the thing with Brett, yeah, really resilient. It is loves to live in barrels, loves to live on wood. And I was just saying at the end there, if you are taking a wine thief to sample out of your barrels, it's good practice. I know it's really difficult to do in the moment, but to clean your thief, sterilize it between taking samples. Just know you will be transferring not just Brett, but if there's other spoilage, yeast or bacteria in there if you are going from barrel to barrel. So just, you know, something to remember. And there is a new product from Lamoth that I just saw, and it's called Kill Brett. And it's not just for Brett. It can be used against other microorganisms as well. There's preventative and curative doses for these infections, so you might want to check it out. It's a chitosan, it's a fungal chitosan, so it has a different mechanism than SO2, and it might be an option for you. I've not used it. I've just seen it a couple times in emails that have come across my desk, but I just wanted to throw it out there as a potential option, and maybe it's something that at the Institute we can figure out a way to trial it. Okay, so moving on, another one that gets talked about a lot, Hanseníasporum uvarum, also called Cloacra apiculate. And reason for that is the lemon shape that it often has. So you can see that in the picture here. It's the main yeast that's found on grapes. So this is what comes in on your grapes after you pick them. So this is almost for sure going to be present on your grapes. They can ferment sugars up to 5 to 6% ethanol. Past that, they're probably going to be outcompeted by the Saccharomyces cerevisiae, as well as the ethanol will start to kill them. But know that they're there, and if you are, for whatever reason, trying wild yeast fermentations, that this may be a contributor. So the cell description, like I said, they're kind of lemon shaped, and that's because when they bud and they split, they bud from each end, and so this is why it causes the shape to become lemon shaped. In wine spoilage, if they are allowed to grow, so if you don't inoculate a high Saccharomyces cerevisiae inoculation and you allow the Hansenia sporum to proliferate, you will get large amounts of volatile acids and esters. So acetic acid and ethyl acetate, will be in higher concentration. And we talked about acetic acid a couple months ago. In small concentrations, acetic acid can help bring a fruitiness and lift some of the other aromatics, but be very, very careful if you are seeing or starting to smell any acetic acid, because then you're getting over threshold, and then you're getting into the difficult to manage area. Okay, next one we're going to talk about Pichia. So there's several Pichia species, membranifasciens, anomala, and fermentans. So they also, a lot of Candida species or Pichia species, they're not necessarily always Pichia and always Candida. They don't always go together like that. But some of the ones that are important for wine are typically Pichia species are a film yeast. So this is the stuff that grows. There was the picture before. I might just back up. This is film yeast, and chances are you might not have seen it quite this bad on your wine, but film yeasts grow a lot. I have them grow on my wines in tank, and in very small, you can just start to see some. It almost looks like dust on the top of the wine. You don't want that. I mean, that's not what we're going for, but they are common. So they need oxygen to grow. So because they really only live at the surface, they probably have slightly less effect than some of the yeasts that are growing within the wine. But that doesn't mean we shouldn't, get rid of them and try to prevent them. There are lots of different shapes in terms of Pichia. You can have spheroidal to elongate shapes, and they produce by different types of budding, polar budding or multilateral budding. So it just depends kind of on the species. They can survive at high ethanol. So that's, they're growing on the surface of our finished wines, right? They can ferment acids as well. And so during, if they are going to cause a spoilage issue, they're producing things like acetaldehyde, which we often consider in terms of oxidation. They can produce acetic acid, ethyl acetate, isoamyl acetate, which smells like banana. So yeah, definitely something. If you see Pichia, if you see a film yeast, skim it off. You could sprinkle some SO2 or KMBS on the top, but definitely skim it off and get it out of there. That's one thing I used to do at a big winery I worked at. We would go around monthly with a little fine mesh kitchen sieve and skim off all the film yeast. You don't want to get to that stage. I realize now how gross that kind of was, and how prevention obviously is key here. Schizosaccharomyces pombe. I wanted to show this because it's in every textbook. It's really classic because of the way that it reproduces. It's got spheroidal to cylindrical shape, and it can ferment some sugars and L-malic acid. The really cool thing, though, is it reproduces by binary fission. So essentially a membrane, which you can see in a couple of these. I don't think you can probably see I don't know if I can get, oh, no, now I'm screwing things up. There we go. A couple of these, you can see like a membrane forming in the middle, and then it just literally separates at some point. And so it's talked to, in academic, classes or whatever, we always talk about it because it's really interesting. In terms of wine, it's actually really important because it has some cap- capabilities that Saccharomyces cerevisiae doesn't. Like it can ferment L-malic acid. So malic acid um, in our wine, we could get, and the researchers have actually grabbed genetic pieces of material out of Schizosaccharomyces pombe and put it into Saccharomyces so that um, it could metabolize L-malic acid. And there was at one point a malolactic yeast on the market, and unfortunately it wasn't really accepted very well, but it has been designed, and they use some genes from bacteria and they use some genes from this particular yeast as well. But in wine spoilage in higher concentrations, it can produce acetic acid, acetaldehyde, and ethyl acetate. And I don't think this is quite last. Maybe this is lastly. And there's a reason it's last because it is probably the scariest one of them all. It is Zygosaccharomyces baeylii. And if I pronounce any of them wrong and you know better, I apologize. But so this one is highly tolerant to sugar, highly tolerant to ethanol, highly tolerant to sulfur dioxide, and other preservatives. It also looks like Saccharomyces cerevisiae. It's spheroidal, ovoid. It reproduces by multilateral budding, similar to Saccharomyces cerevisiae. So it's really hard to identify unless you're using genetic methods or and it can grow very well in wine, and it can metabolize a bunch of things. It can metabolize malic acid as well, but it can grow so much that it can lead to turbid wine. So if your wine was totally clear, ready to go, and then all of a sudden it went turbid, be worried about this. The other thing about this is it's hard to get rid of once you have it. It's very tolerant to cleaners as well. It can be sensitive, however, to peracetic cleaners as well as quaternary ammonium cleaners. And it's sensitive to hypochlorite cleaners, but we're not going to bring bleach into our wineries, right? And so yeah, it's persistent and it's very hard to get rid of. And if you have an infection, I would be very concerned for the winery that there's other places of infection and that it will infect future batches of wine. So this is one that you kind of, well, I'll get to what you should do if you have infection. But this is one that you probably do want to get some sort of identification if it's Zygosaccharomyces. So I did want to mention a couple of, oh, and the rest of my slide didn't get saved. I apologize for that, but I'll talk about a couple things. But spoilage bacteria and botrytis, because these, I didn't want to leave bacteria out completely. Botrytis is also a mold that we can see, and it infects our grapes. So the main two categories for spoilage bacteria, we've got our lactic acid bacteria and our acetic acid bacteria. And I want to point out too, we need to be really thankful that we work in wine that has higher ethanol concentrations and lower pH, so that number one, pathogens cannot live in wine, and number two, there's a much smaller subset of species that can actually infect our wine because of high ethanol, low pH. It really gives us a leg up. And so lactic acid bacteria, some of them are good, like Enococcus eni, but a lot of them are bad. And so we can get things like geranium taint, diacetyl taint, mousy. So there are some faults that we do associate with lactic acid bacteria. Acetic acid bacteria, you're going to get mostly your acetic acid, acetaldehyde, ethyl acetate. And they use a lot of different food sources. So acetic acid bacteria can use acids, but also gluconobacter there can use glucose to turn it into acetic acid. So we don't want that. So we do want to keep our bacteria in check as well. Thankfully, some of the techniques are the same for yeast and bacteria. Botrytis, like I said, I wanted to mention because it is a mold. It can come in on grapes. And oh, well, I'll get to that in a second, of how to field sort and whatnot. But be aware of it. I mean, I don't think anyone here in the Midwest is doing any noble rot. I think that's really, we don't have the climate to be doing noble rot in. The last thing I wanted to point out too, another fungus that we, and it's not a specific fungus, but a group of fungus can live on corks, and that's how we can get cork taint. It's not the only way you can get cork taint from trichloroanisole, but fungi can live on corks and metabolize some cleaning agents and whatnot that it probably came in with to give you trichloroanisole, which is cork taint. So let's just talk about prevention, because I think that's the key here to avoiding these infections with these yeasts. Use clean, healthy grapes. And how do we get clean, healthy grapes? We have a good integrated pest management system going during the growing season. So that includes your sprays. That includes canopy management to be allowing airflow and whatnot. And it also includes scouting of your vineyards to be, to actually be visually looking and seeing if you see any molds or mildews or sour rot issues or things like that. You need to be in your vineyard and looking for these things as well. Sorting, you can sort in the field or at the winery. And so what we tell the hort farm when they're harvesting our grapes, any grapes that look damaged, severely damaged, or smell like acetic acid, don't put them in the lug, and don't bring them to the winery. Basically, you could also cut out pieces of a grape cluster if that would help clean it up. I know that some of you growers get paid by the ton, but be scrutinous as a winemaker too. And if you're using grapes from your own estate, just you want to be starting off with the best possible grapes without bringing any extra microorganisms in. There's lots of yeast and bacteria that live on grapes, but we want to minimize the bad ones. If you're storing your grapes for any length of time, I would highly recommend that before processing, I'd highly recommend that you spray the top of the bins or the top of the lugs with an SO2 solution, and that'll just keep other things at bay. And it will keep, yeast that are growing on your grapes, if anything got damaged, they're not going to start kicking off fermentation, okay? In the winery, keep your winery clean. This is really, really super important. Your winery needs to be a very clean space. Stuff can cross-contaminate very easily, and so don't allow that to happen. Ferment quickly after processing. I didn't really realize this. I guess on the wines that I make, I do a 24-hour cold settle and then rack and inoculate right away. I know some people are doing longer cold settling, and that may be required for your particular cultivar, and that's fine. Just be careful. Make sure you've got SO2 in there to prevent wild yeast fermentation. Maybe that's what you want. But if that's not what you're going for, I would highly recommend a 24-hour cold settle, rack and inoculate to prevent anything from kicking off that you're not inoculating with. I would highly recommend a co-fermentation for malolactic fermentation. So I've done this for several years now. You inoculate 24 hours after a yeast inoculation. There are specific strains of bacteria that you can use. And what this does is it prevents, at the end of your primary fermentation, if you're going to go on and do a, what's it called, a malolactic fermentation, you don't want that's a long time to not be protecting your wine with SO2. And so I'd highly recommend that you do a co-fermentation and get those two done together as much as possible, and then protect your wine with the SO2. Protect your wine with SO2 as soon as your sugars, malic, are finished. Make sure that it's based on pH, and remember that that first addition is going to have a significant amount of it bound up. So make sure that you are taking that into consideration when you're deciding how much SO2 to add. So 40 to 60% of that initial SO2 add will get bound up. And like I've said in the previous Lunch and Learn, you don't want to be chasing your tail to keep adding free SO2. Minimize headspace and gas any headspace with inert gas. So you can use CO2, argon, nitrogen, gas your tanks before you go into them. And this is going to prevent a lot of those film yeast. Check your large tanks regularly for film yeast. Check your variable capacity lids. And as Drew Horton will probably say at the end of this, don't just check the gauge, that the gauge says that it's in the green. Physically go and tap on that lid and make sure it hasn't, there's no issues with the gauge not reading what's actually happening with that gasket. And these things take a quick amount of time compared to what I'm going to tell you in terms of if you have an infection. Check your SO2 and acetic acid levels regularly, and taste regularly. Your taste buds and your mouth, your own mouth, is a really good instrument to pick up if you've got any infections going on. Finish your wines quickly and don't age them. If you've got wines in a tank, a stainless steel tank, and you think you're aging them in tank, you're not doing too much. And so I highly recommend you retain the freshness and get those into bottle. They are much safer in bottle than they are in a large batch in a tank. If you're doing oak aging or something like that, that's a different story. But even with that, once you're finished, don't just throw them back into a tank and leave them sit for another 6 months to a year. Get them in bottle. Get things in bottle as quickly as possible. That will prevent infections. Okay, if you have an infection, what should you do? A couple of things that you may be able to do: check your SO2 levels and adjust to an appropriate, and this needs to be based on the pH, okay? So if you are at a pH of 3.5 or above, you're going to need a lot of free SO2 in there. I don't have the chart on me, but you're going to need a lot of free SO2 in there to protect your wine. Find and filter that wine to remove your spoilage yeast. You have to get it out of there. Filter to 0.45 micron to ensure that you get yeast removal. You want to filter to 0.2 micron to ensure bacteria removal. That's really hard to do that. Because most of us, first of all, it takes a long time. We can get pressure backups and things like that. So you really want to prevent these bacterial infections, because even when I'm bottling my wine, I don't go down to 0.2 micron. Something we do in the lab, but we don't necessarily, it's not very practical to do in the winery. Recheck your SO2 after filtering, okay? Make sure that no wine chemistry went awry during the filtering process. And you can send away for analysis to be sure you remove the yeast or bacteria. So you can send away and ask for the analysis I've done before. It is called a scorpion panel, and you can ask for different, just an overall yeast count or whatnot, and they can, you can get some insurance, I guess, whether you removed your yeast or bacteria that you were worried about. Okay. So depending on the amount of wine and the type of the infection or the fault, you could blend with another wine that's fault-free if it wasn't such a huge issue in terms of sensory impact. You could blend with another wine. After you blend it though, please check your SO2 and please check your stability, your heat stability and your cold stability. As soon as you blend wines, you can throw those things off. So just because two wines were stable to begin with, if you bring them together and blend it, doesn't mean they're stable afterwards. So make sure that that's a check that you do. You can distill it for fortified wine or for cleaning or things like that, but you can distill the wine and get some use out of it. And you can dump the wine if it's really that bad. Yeah, that is an option, right? So you don't have to sell bad wine. You do not have to sell infected wine. I know it's a loss in terms of money, but think about the long-term ramifications of selling bad wine to people. Clean, sanitize, and sterilize your tank and any other vessels, hoses that infection came in contact with. You do not want to infect the next batch, okay? So if you didn't do prevention at first, you're having to deal with an infection, you sure as heck want to, prevent for the next time. And then depending on the type of infection, you may want to have analysis done on other wines or swabs of other winery locations. So if you think for some reason, or if you attack your infection and you get it under control and you adjust the SO2, you filter it, maybe you blend it out, you do whatever, and you're feeling good about the way that you took care of it, that's fine. You probably don't need to know what it is that it was, what the infection actually was. If you find that you are continuously having problems, and maybe even in the same wine, you're continuously levels are keep going up for acetic acid or something like that, you may want to send out for identification. So I would say, you go in after it, you try and take care of it once, and if you at that point, if you're still having problems, you're still finding that the wine's increasing in whatever fault it is, or still having a haze or something, creating a haze or something like that, send it out for, because if it is something like Zygosaccharomyces, you want to know because it's probably all over your winery and you probably need to do a serious deep clean. So, so don't wait too long if to get identification. Okay, so then in conclusion, and we can talk about things, I can answer some questions in a second here, but in conclusion, protect your grapes, protect your wine, okay? Prevention is key. Regularly walk your vineyards. You don't want to be bringing bad stuff into the winery, and regularly taste and test your wines. And if there's something you can't do or you don't know how to do, please call us. We've got protocols. We can do analysis for you, but we'll also walk you through how to do things in your own winery. Identification after the first attempt of control, because it can lead you to be doing things that are more specific. If you have a specific yeast infection and yeah, there could be better options for control in terms of specific yeast. SO2 is important. We want to make sure that that is at a good level in our wines, but remember, we don't want any oxygen exposure. Because a lot of those yeast like oxygen. Find and filter your wines and get those things out of there quickly, and bottle your wines quickly. No need to age them in stainless steel tanks. Okay. So that's kind of it for the discussion on yeast spoilage, or spoilage yeast, I should say. Our next Lunch and Learn will be held February 5th. We're taking a break in January. So February 5th, and watch out for a list of the topics. If you have any ideas, please let me know. But watch out for a list of 2024 topics. The reason we're taking a break is because the Iowa Specialty Producers Conference is that first Monday in January. Actually, the first Monday is is New Year's Day, so that's really why we're taking a break. But the Iowa Specialty Producers Conference will be in Ankeny, where it usually is at the FFA Center. We've got a good program lined up for you, so consider coming to that. And I also wanted to, this isn't out yet, Anne might get mad at me, but there's a new podcast coming from Randall Voss and Anne Zwink, and I highly recommend once it drops, is that what we say in, is that what you say in the podcast world? When that drops, please check it out. Randall and Anne are going to have some interesting discussions with winemakers, wine judges, grape growers, the whole gamut of people that are associated with this industry. So I think that's going to be a really fun, and it's something fun, I think, to probably be listening to while you're working or that sort of thing.
Update on
And then I
that.
also wanted to let you know about a couple webinars that there's one coming up December 13th. So I encourage you to Eastern Viticulture. I didn't spell that right, but or the acronym. Just Google that. It's from Penn State University is technically putting that on, but it's all including Iowa State that is involved in that webinar series, and that's starting back up December 13th. And then ASCV Eastern section does a hang time series, the third Wednesday of the month. I don't know what the topic is going to be for this month, but please Google that if you're interested. It's for industry, and they've covered a lot of interesting topics so far. I just wanted to bring that one up. Oh, unwrapping wine. And what did, okay, hold on. I'm going to stop sharing my screen here in a second. Anne had a, I think it drops today. I think it drops today, the new podcast. And you can find it on, well, I think you can find it on YouTube. Anne, do you want to actually, before, is it on YouTube, Anne? Before we stop recording?
Can you guys hear me?
Yep.
Okay. It's on a